Good morning, everybody. Today we are going to start nutrition in human beings. As you can see that I have already made a chart of nutrition in human beings. Nutrition in human beings occurs only when we have the human digestive system. Human digestive system has two parts. One is different types of organs and the other one is glands. The human digestive system organs consists of mouth, or we can call it as buccal cavity, esophagus, stomach, small intestine, large intestine, anus. It also consists of some important glands like salivary gland, liver, pancreas. Apart from these three glands, there are many other glands which are associated with these organs, which we will be reading in the course of time. Now we will start with the first we'll start with the organs. So the first organ that we are having with us is the buccal cavity or mouth. The buccal cavity has two important structures, as we all know, that is the teeth and the tongue. Along with that, it also has the salivary gland. Now, the teeth, it helps in making the or breaking the food into small pieces by the help of the teeth, like incisor, in break into small pieces by the help of incisor, canine, premolars, and molars. Tongue helps in mixing of saliva with food. Saliva mixed with food. So this process in the tongue helps in mixing of saliva with the food, and the salivary gland secretes saliva in which there is an enzyme which is called the salivary amylase. Enzyme which is salivary amylase. When the saliva is mixed with the food, the salivary amylase digests the when the saliva is mixed with the food, the salivary amylase digests starch. In the presence of salivary amylase, is digested to form sugar. So the starch is a complex material and sugar is a simple material. So in mouth or in the buccal cavity, the salivary gland secretes salivary amylase, which digests or breaks down starch into sugar. Now this we can also do it experimentally. Also we can prove this. There is an experiment in your book which is experiment 6.3. In this experiment, what we need to do is we will take two test tubes. This is test tube A and this is test tube B. Now in both these test tube we will take what? Now starch solution of required amount, like for example 1 ml is taken in both of them. So this is starch solution is taken of 1 ml. Now what we will do, we will add 1 ml of saliva, then we will add, this is first thing, then second thing, we'll add 1 ml of saliva from our mouth to test tube A. We'll add 1 ml of saliva to test tube A. Now this test tube has 1 ml of saliva added to it, and we will keep this thing for around 20 to 30 minutes. It is hold on or kept for 20 to 30 minutes. Then after 20 to 30 minutes, we will add dilute iodine solution to both of them. We'll add how much? Few drops of dilute iodine solution to both. And our observation will be test tube A will be showing a red color of iodine and test tube B will be showing blue black. This is the observation. Now why does this happen? Test tube A shows red color of iodine and test tube B shows blue black. As we all know that starch reacts with iodine or iodine reacts with starch to form a blue black color. But in test tube A there was no blue black color. That means starch was not there. That means we have added starch in both of them, But what extra we have done is we have added saliva in test tube A. So saliva may have digested the starch present in it, due to which there is no starch left over, and that starch is converted to sugar, and iodine only reacts with starch and not with sugar. Hence the iodine did not produce any blue black color. Rather, iodine was showing its own color, which is red or reddish deep red color, which was showing its own deep red color and was not converting itself into blue black. While in test tube B, we have not added any saliva, hence the starch was as it was present. There was no change in its structure, neither it was broken down. Hence, when iodine was added to it, then both of then this iodine solution reacted with the starch of test tube B and produced the blue black color. This gives us experimental proof that starch in the presence of salivary amylase breaks down to form sugar. Now this happens in the mouth. Then the second organ which we will be going through is esophagus. This is the second organ. In esophagus, there is no digestion. There is no digestion, but from here, throughout the gut, what happens? Peristalsis occurs. Now, what is peristalsis or peristaltic movement? It is a wave-like movement. It is a wave-like movement like this. Then the food will be pushed down, so downwards it will be swelling. Then the food will be pushed down, so downward it will be swelling. So this is a peristaltic movement which occurs in the esophagus, so that the food is pushed down to the next part, and the next part is what? Stomach. Now in the stomach, when the food arises or when the food arrives, then the food enters into the stomach, and in the stomach, the inner lining of the stomach has muscular walls. Inner lining is made of muscles, or the muscular walls carry out what? Expands and contracts for mixing of its juices with food. Now, what are the juices of stomach? The juices of stomach is produced by a gland called gastric gland. Now, this gland is associated with the stomach only. Now, this gastric gland has three things associated with it. One is HCl. Second is pepsin. And third is mucus. Now, HCl's work is to provide acidic medium for activation of pepsin. So, pepsin is a protein digesting enzyme which is present in the gastric gland of the stomach. This always remains in the inactive state. When HCl is secreted, it provides an acidic environment for the activation of the pepsin. And when pepsin is activated, it will cause digestion of protein. And there is the next one, which is mucus. Mucus, it acts as a protective layer because of and protects the inner layer from the action of the acidic HCl. It also acts a protective layer for the action against HCl. So it acts as a protective layer for protecting the inner walls from HCl. HCl not only provides acidic medium, it also helps in killing of germs. It also helps in killing of germs. So in the stomach, the muscular wall of the stomach expands and relaxes, or expands and contracts to help in mixing of the food, mixing of the digestive juice with the food. And this digestive juice is secreted by the gastric gland present in the inner wall of the stomach. This gastric gland, which is present in the wall of the stomach, releases HCl, pepsin, and mucus. The HCl helps in making the environment acidic so that the protein digesting enzyme, which is pepsin, gets activated. Then this activated pepsin helps in digestion of protein. The next one is the mucus. The mucus helps protects the inner wall of the stomach from the action of HCl. HCl not only HCl not only activates pepsin but also helps in killing of germs. So these are the functions of inner wall of the stomach and hence together. Now we will move on to the next factor, which is the food exit out from the stomach, and the stomach, as we know, is a J-shaped organ. Let me draw a proper picture here. This is a J-shaped organ, and the upper part and the lower part, the entry and the exit, is guarded by sphincter. We call it as a sphincter. Here also exit is also guarded by a sphincter. Now when the food after digestion, semi-digestion, comes out from the stomach, the entry and the exit is also guarded by a sphincter, and it reaches the next organ, which is organ number 4, that is small intestine. Now small intestine is a place which is the largest or the longest part of the body, and it is shows extensive coiling. Therefore it is situated in the small part, and the small intestine is longest in case of herbivores and smallest in case of carnivores. The reason is herbivores carry out digestion of cellulose, which is not carried out by carnivores. Hence the small intestine is longer in case of herbivores and smaller or shorter in case of carnivore. But if we compare the whole body organs, then small intestine is the longest organ. Then let us move into it. It is a site in which complete digestion occurs. So the site of complete digestion is the small intestine. And in the small intestine, there are two things which are associated with it. Now into the small intestine, three things comes. First is the semi-digested food. Semi-digested food from where? From stomach. Semi-digested food from stomach. Second, bile or bile salts from liver. So liver also gives its secretions to the small intestine. And third is pancreatic juice from pancreas. So liver gets these the small intestine gets these three things. But the problem is the food which we are getting from the stomach is which type? It is acidic because of the action of HCl. Now hence the food has to be made basic. So the first thing which happens here is acidic food is converted to basic form, and this occurs because of the action of bile salts. So bile salts convert the acidic food to basic food, and this basic food formation of this basic food helps in the action of pancreatic enzymes. So whatever pancreatic juice or enzymes come from pancreas, they will only work on a basic food, and acidic food it will not work. So bile salts convert the acidic food to basic food so that pancreatic enzymes can work on it. Along with that, while converting it to the basic food, also another bile salts do one more thing, that it also breaks down larger globules, larger fat globules. It breaks down larger fat globules. In the presence of bile salts, it will break down into smaller fat globules. It is just like you have read about emulsification. In emulsification, what happens is we are removing or breaking down larger fat molecules by addition of a salt. So here the name of the salt is bile salt. This bile salt, it will break down larger fat globules into smaller fat globules, and this process also is known as emulsification of fat. Now after this happens, the pancreatic juice will act on both of them. Now third is the action of pancreatic juice. Now pancreatic juice has some important constituents, like The pancreatic juice contains the first important thing, which is trypsin. This trypsin helps in digestion of proteins. So whatever semi-digested protein is there, semi-digested protein is completely digested. Semi-digested protein is completely digested by the help of pancreatic enzymes. Similarly, it also has an enzyme called lipase, which acts on smaller fat globules, digested to smaller fat globules. This acts on the smaller fat globules. Another is pancreatic amylase, which acts on sugar and other leftover starch or other leftover complex carbohydrates. So finally, what happens in the small intestine is the proteases plus peptones break down to form amino acids in the presence of trypsin. This is point 1. Point 2, small fat globules break down in the presence of lipase to fatty acids and glycerol. And third is carbohydrates are broken down in the presence of amylase to glucose. So hence digestion is over. Now digestion is over. Now you must be thinking that what is the function of then large intestine. Then the next organ which we are going to start is the large intestine, which will be organ number 5. So large intestine. It is a place where absorption of digested food occurs. Food and absorption of water. Actually, in the small intestine also there is absorption of digested food, but some leftover digested food and some vitamins which are soluble in water are digested in the large intestine. And the major function of large intestine is absorption of water. So whatever water is there, it is being absorbed, and this occurs because of the presence of villi. Villi are finger-like projections which increase the surface area of our absorption, which increase the subsurface area for absorption. So finally the absorbed, everything is absorbed. So what is left behind? Left behind in the rectum, the left behind ones is undigested food, which is thrown out by anus, which is organ number 6, which is thrown out by anus. So anus helps in removal of undigested food. Now see this line in the book: the unabsorbed food is sent to large intestine where more villi absorb water from this material. The rest of the material is removed from the body via anus. The exit of waste material, this is an important point, the exit of waste material is regulated by anal sphincter. So here also in the anus there is a sphincter which is known as the anal sphincter, which regulates the exit of waste materials.